Well, we will go with the final presentation now. I thank you all very much to keep staying here, although the presentations are pre-recorded, some of them. So we will go now with the final presentation of Josefina Cruzat. Josefina Cruzat is a lead researcher in the Latin American Brain Health Institute in Chile as well, and she will present social determinants of brain aging, a multimodal approach to brain age gap and its mechanistic underpinnings. Josefina is a PhD in theoretical and computational neuroscience, and she will present something similar, mechanistically speaking, of the presentation of Carlos, but from an MRI and fMRI perspective. Thank you. Thank you, Josefina.
Hello everyone, I'm Josefina Cruzat from the Latin American Brain Health Institute at Universidad Adolfo Ibáñez in Chile. I'm sorry I couldn't be there with you in person, but I'm grateful still for the opportunity to share our recent work on how we combine multimodal neuroimaging, machine learning, and whole brain modeling to study the brain age gap and how this biomarker can help us understand the impact of social determinants of brain aging, especially in underrepresented populations such as Latin America. Aging is one of the biggest biomedical and social challenges of our time, not only because it's responsible for this huge demographic shift that we are experiencing worldwide, but also because aging is the leading risk factor for nearly every chronic disease, from cardiovascular to metabolic to including neurodegeneration, cognitive decline, cancer, diabetes, right? And we know that aging doesn't follow the same path for everyone. Some people stay cognitively sharp until late life, while others decline earlier and faster, so showing that and meaning that the pace of aging could be highly variable, and this variability has driven a shift from measuring aging by years lived to study now how fast or slow different biological systems age, including, of course, the brain. In recent years, the concept of brain clocks has gained a lot of attention among the neuroscience research community. These brain clocks are models that estimate how old the brain appears based on neuroimaging data. And these models allow us to move beyond chronological age and ask questions like how old does the brain look, or in terms of functionality or structurally or structure, and this allow us to calculate the difference between the predicted age and the actual age, and this is called the brain age gap because it could be different, right? So a positive gap is when the brain looks older than you expected by your chronological age, and this has been linked to cognitive decline, neurodegenerative diseases, and even increased in mortality. By the contrary, we have a negative gap, that is where your brain appears much younger than is expected from your actual age, and this is associated with more resilience, better outcomes in aging, better functionality in general. And there are even more and more studies on this topic, and some of them have shown that this brain age gap captures very important clinical and social information. For instance, there is a study with the UK, using the UK Biobank data. They show that the brain age gap correlate with genetic risk and brain atrophy. And in Latin America, work using the Red Lab Consortium data has shown that this brain age gap reflects socioeconomic disparities in brain health. Well, but the thing is that despite all this clinical and social relevance, there is still a major gap in this research area, which is that we still don't fully understand why some brains age faster, what are the neural mechanisms that drives this gap. This is because most existing models are descriptive, so they predict the brain age gap, but they don't explain it. And on the other hand, these models often rely on a single modality, and mostly of them focus on the structural aspect. And they overlooked all the functional dynamics of the brain. So in our study, we aim to bridge this gap and to go one step further by integrating structural and functional imaging with whole brain modeling to explore the mechanisms behind the brain age gap, and to explore how these gaps are shaped by social determinants of health, and they associate with cognitive capacity. So our main goal was to understand what drives the brain age gap, not just to predict it, but to explain it. And we wanted to uncover the biological mechanisms behind it and to explore how social and environmental factors might shape those mechanisms, especially in populations that have been historically underrepresented in the neuroscience research, such as Latin America. And our hypothesis builds on 4 key ideas. First, that chronological age and biological brain age are not the same. Some individuals, of course, age faster and others more slowly. Second, that brain diseases like Alzheimer's disease and frontotemporal dementia reflect a form of accelerated aging. Third, that brain age is not only genetically determined, but also shaped by lifelong exposure, including environmental and social factors. And finally, that the biological pathways driving brain aging are themselves influenced by context. So, meaning that where and how you live affect how the brain ages. And to do this, we analyze data from more than 1,800 participants across the cognitive spectrum, from healthy controls to people with memory complaints, mild cognitive impairment, Alzheimer's disease, and frontotemporal dementia. Our samples came from both Latin America and the US through the RedLat consortium data, the ADNI, and the LONI databases. And this was very important because this allowed us to capture not only the clinical variations in terms of this cognitive spectrum, but also this regional and socioeconomic diversity. And with this data, we trained 2 support vector regressors, one using gray matter volume from structural MRI, and the other one using functional connectivity from resting state fMRI data. And we then combined the outputs of these 2 models using this model stacking with rich regularization to generate one simple and more accurate brain age estimate. And from there, we computed the brain age gap and corrected for potential biases, adjusting for chronological age. Then, to explore the underlying biological mechanisms of this brain age gap, we used a dynamic mean field model, which is whole brain biophysical simulation framework that estimates 2 key parameters: the global coupling and the local feedback inhibition. And these parameters were based on each participant's own empirical connectivity and atrophy data. And finally, we examined how the brain age gap correlates to cognitive performance and also how it is influenced by social determinants of health. And for that, we used a composite social determinants of health index that captures different aspects like education, income, housing, and access to care. We found these are the results, And we found that the model performed well in predicting brain age among cognitively normal adults. Then we found that the model improved accuracy when we combine these two data, structural and functional, compared to the performance using unimodal or traditional machine learning models. And also we showed that our model reduced prediction error and explained more variance across clinical groups and regions. And this suggests that to estimate brain aging, especially in diverse populations, it is better to move beyond structure alone to integrate multimodal signals, such as functional connectivity. And we also examined which brain regions contributed most to the predictions. And we found that structurally, higher brain age gaps were linked to lower gray matter, especially in the hippocampus, thalamus, and posterior cingulate areas that are areas which are key for memory and for information integration. And at the functional level, we found that the bug was associated with a weaker connectivity across networks that involve the hippocampus, insula, and frontal regions, which are key areas that are relevant for emotional and processing and for executive functions. Then we applied our model across the diagnostic categories, and we saw what we expected, right? That the brain age gap increased with disease severity. So Alzheimer's disease and frontotemporal patients had the larger gaps, followed by mild cognitive impairment and subjective complaints. And finally, the healthy controls. And here, what is this very surprising, striking, and sad is that cognitively normal adults in Latin America had significantly higher or larger brain age gaps compared to their counterparts in the US. So, again, healthy controls in Latin America appear older than healthy controls in the US. We next ask how brain age gap relate to cognition and to context, right, to the social determinants. And we saw that among patients, a larger gap was linked to lower cognitive scores, confirming the clinical relevance. And we found that in cognitively normal subjects from Latin America, there were higher brain age gaps associated with greater exposure to social disadvantage conditions, measured through this composite marker, suggesting that brain aging is not biologically determined, but also it's a marker of long-term exposure to inequality and different risks. Then, to understand why some brains age faster, we need to move beyond correlations and look at the mechanisms. And for this, we used a whole brain biophysical model, the dynamic mean field model I already introduced, that simulates the neural activity across brain regions by capturing these two key processes: the strength of communication, which is the global coupling parameter that reflects how well the different brain regions interact among each other, and then the second parameter, which is the balance between excitation and inhibition, which reflects local control. And what we did is that we compared these dynamics between people with high and low brain age gaps across both, again, regions and clinical groups. And here's what we found. People with higher brain age gaps showed two patterns. First, they had lower global coupling, meaning reduced large-scale communication across the brain. And second, they had increased local inhibition, especially in regions that were affected by atrophy. So this points to a shift in network dynamics, possibly reflecting a compensatory response or an early marker of dysfunction. And again, these patterns were seen even in cognitively normal individuals with high brain age gaps in Latin America, particularly there. So in summary, our findings shows that brain aging is not only driven by biology, it's also shaped by social inequality. Even cognitively normal individuals from Latin America show signs of accelerated brain aging compared to those from higher income regions. This was linked to reduced brain connectivity and signs of increased neural inhibition. And finally, the results highlight the need for early and more equitable interventions, especially in underrepresented populations. This to promote healthy brain aging and address all the disparities we have in dementia risk. And with that, I want to thank you so much for your time and your attention. I am grateful to my collaborators and participants across Latin America and the US, and I hope you enjoy the rest of the conference.
Okay, well, thank you very much for assisting the symposium. Thanks for the speakers, and special thanks to Jess that she's here. and I hope you enjoy the rest of the conference. And any question about the institute that we have in Chile, or the collaborations, or some specific things about the work that was presented, I will be here all the conference, and Jess will be here as well. And the emails of the authors were in the presentation, or you can ask me directly for their contact. Thank you very much.